me just give a quick introduction. So Martin and Anna are working with Novartis. Anna specifically has been with Novartis since 2006, where currently her role is as an associate director and senior principal data scientist in the Global Discovery Chemistry Department. There she works for hit finding technologies and hit to lead optimization. Martin is a structural biologist with several years of experience in chemical biology and drug discovery. And currently he works as an innovation fellow in the chemical biology and therapeutics department at Novartis with a focus on structure guided drug design. Looks like maybe we're having some slide technical. Here we go. Perfect. And today they'll be talking about some of the recent work from Novartis on reversible DCAF1 ligands. And with that, I will hand it over to you two.
Hi, yeah, sorry for the technical hiccup. It's a classic thing in this modern age. Super happy to be invited. Thank you so much for the organizers. Very nice that we get the chance to present our work on a novel ligase, such as Soren mentioned that he didn't have time to talk about. So we have luckily a bit of time to talk about our findings on DCAF1. It's not the most novel ligase, but we will take the opportunity of the seminar to walk you through our work on how we discovered new reversible ligands for DCAF1 and turned them into PROTAC designs. So with that, we can go to the next slide. So first of all, I want to acknowledge that this was a lot of people. This was a massive team effort over a couple of years here at Novartis, and I want to highlight both Claudio and Martin, who spearheaded the project together with Anna here, and then on the late stage, I joined to bring it over the finishing line together with Nico Tome from the FMI. A lot of different departments here at Novartis helped. All did tremendously great work, and yeah, just as a brief starter to all of this. So I think why we should look into novel ligases is a bit of an obvious question to this audience, but I will just quickly summarize a couple of points. So I think new ligases bring new opportunities in targeting targets that cereblon can't do as well, or VHL doesn't do it either, as well as potentially finding ligands that are drug-like, similar more to cereblon, less than VHL. And also, maybe we can address some of the tox issues that might occur if you use cereblon, or by getting ligases that are more tissue specific, or even subcellular location specific to the target of your interest. And the other thing that is quite interesting, and I think it was recently highlighted by work from Georg Winter's lab, very nice, is resistances to cereblon-based PROTACs as well as VHL-based PROTACs. And as you can see in the graph that I took from their publication, the non-essential ligase cereblon in their studies mutated on a much higher rate than the more essential ligase VHL. And I think this really sets the scene well for why you look for new ligases, because this, if you look for more essential ligases, you can probably address this upcoming resistance in cancer patients in a better way. And with that, we can move to the next slide, please. So when we looked at the portfolio of ligases that are out there, there's obviously over 600 of them. So you have to make a choice of which one you want to work. So first of all, we decided, well, we need to find the ligands. So we need to find a target that you can easily target if you find ligands for with a high potency. And therefore, we focused on WD40 containing E3 ligases. From in-house data as well as published data, it was quite obvious that you can develop highly potent ligands to those domains. So we had a look in terms of E3 ligases that contain one of those domains and ran them through the Demeter pipeline to see which of them turned out to be essential in this experimental setting by the Broad Institute. And you can see that there are a couple of outliers here highlighted. On the left is DCAF1. Others are containing essential genes such as DDB1 as well. And if you focus on the right side of the slide, we selected ligases that had been known to work in TPD at that time when we made the figure. Now there are a couple more to add. But back then, it was quite striking that within that data set, DCAF1 was a stronger outlier than, for example, VHL1. And the other thing that helped us to really boost this was upcoming publications in the field that highlighted, well, you can target DCAF1 with covalent ligands that can be turned into PROTACs. So all of this gave us confidence that DCAF1 is an essential ligase. It has a domain that we can likely target, and therefore we will use this to look for ligands that can be turned into PROTACs. So DCAF1 is an inactive tetramer. Lab from Nico Thomas' lab really nicely showed that upon nedylation, this inactive tetramer that is shown here as the EM map on the left side turns into an active dimer that is shown on the right side. And the WD40 domains that I mentioned before are highlighted here in orange. Yes, so it's a bit more complex in activation than cereblon, but nevertheless, that encouraged us to go to the next slide. And I think there Anna will take over.
So here I'm showing again the WD40 domain of DCAF1, and the WD40 family. It's one of the largest protein family, about 1% of the total cell protein. And typically it acts as a scaffold for binding to other proteins. And it is made by 6 to up to 8 repeated units called also blade that you see here colored in different colors, which form this beta propeller structure. And each blade is made by 2 antiparallel beta strands. So at the beginning, what we did, we analyzed this structure by using 2 in silico pocket identification tools, sitemap and fpocket, which identify 2 pockets, one in the middle of this beta propeller structure that we call Dunnett hole, and it's highlighted there in white with the white spheres, which are the location in the pocket which are more buried. And this pocket was associated with a good drugability score. And then the second one here highlighted with the black spheres, it was found instead much with a much lower drugability score. And we call it a blade region because it's sitting between the blade 1 and blade 2. So then what we did, we collected a small focus set of WD40 known binders from our internal company knowledge, and we screen those for a total of 21 compounds with the ligand observe NMR screening method against DCAF1. And we found 2 hits, and one is shown here, compound 1, which was coming from a previous EED Novartis project. And this compound 1 was also confirmed to be a true binders for DCAF1 through validation of 2D protein observer NMR experiment using carbon-13 selectively label methionines. And here you see the 2D spectrum in the middle of the slide, where you clearly see protein chemical shift perturbation upon addition of the compound. And but the compound is quite weak against DCAF1. It showed KD greater than 500 micromolar. And despite the low affinity towards DCAF1, we succeed in solving the X-ray structure in DCAF1, and this is shown on the picture on the top right. And we observe 2 orientation in the X-ray structure, which are overlaid in the picture. And in both cases, the imidazoline ring, which is protonated, it makes either an interaction, an hydrogen bond interaction with the carbonate of threonine 1097, or in the other orientation, the green one, it picks and makes an hydrogen bond interaction with the side chain of the aspartate 1356. And we had also the X-ray structure of the same compound solved with the EED, which is depicted in the picture below in orange. And this is a third different orientation, and in which is the phenyl of this 3-cycle core that is pointing more in depth into the donut hole of the ED cavity. And the fact that they assume different conformation between the DCAF1 and EED is not too much surprising, because the WD40 family has a conserved fold, but they share very low sequence similarity. And in fact, the sequence identity between EED and DCAF1 is less than 10%. So then what we did, we explore further this finding, and we collect close analogs of this IT, and looking through 2D similarity and 3D similarity, and looking in our Novartis archive. And we collected an additional set of 30 compounds we screened by NMR. And here I'm showing compound 3 and 4, which showed an improved affinity towards DCAF1 in the 2-digit micromolar range, still keeping the affinity towards EED. And we also solved the X-ray structure of these two compounds, and you see the them overlay on top of each other on the picture on the right. And these share the orientation 1 of compound 1 that I described before. So they are making the hydrogen bond contact with this carbon here of the threonine 1097. And then there is this group on the right, which is more 3D shape, which occupy better the subcavity there on the right, and this explains the better affinity. But we also found that these two compounds also in the blade pocket, this is the picture below, in two different orientations. The contacts are less productive, but then the analysis of all this X-ray structure in DCAF1, but also the compound 1 EED, then suggests the structure-based design strategy to apply. So, which we decided to first grow towards the direction indicated here with the red arrow. First, because we were looking for an exit vector for then developing a bifunctional degrader, but also because through this way, we thought that we could remove the binding to EED as well as to the blade pocket due to the clashing that this would produce. And then we also, the other thing we wanted to explore is to go a bit and grow a bit deeper into the donut hole of DCAF1, and for that reason we morphed the imidazoline ring and we open it in order to reach the contact with this aspartate that we have seen with the post 2 of the compound 1, as I previously described. And so here it's with few compounds I'm highlighting what I just described. So with the compound 6, this is the growing, so the addition of this piperazine, which is tolerated and further improve a bit of the potency. With compound 7, we have morphed this imidazoline, and we have opened it, and we have instead append this amine ethyl chain in order to grow further deep into the cavity, with also some improvement in potency. Then you see compound 8, which is the combination of these two modifications with a KD of 5.6, and we also saw this X-ray structure of it, which is in purple shown below, and these highlight the role of this chain that we added, which indeed depict the contact with the aspartate, the side chain of the aspartate 1356, but also pick two additional contact with the carbonyl of threonine 1139 and histidine 1140. And interestingly is to observe that the NH connected to the quinazoline ring is not really mimicking exactly the same hydrogen bond contact that the imidazoline ring makes itself, but it's another different carbonyl that it picks. And then we change the piperazine into an acetyl piperazine towards compound 9 with an IC-KD of 1.5. And then next, what we explore is the benzylic position in order to feel better that sub pocket there. We grown from cyclopropyl to cyclopentyl with compound 11, and then with compound 13 with cyclohexyl. And in the picture on the right, this is the compound 13 solved by X-ray structure, and also highlighted the role of this acetyl that we added, which is also making a water-mediated interaction. So in short, we started with this fragment quite weak, greater than 500 micromolar, and more potent towards ED. And through this optimization process, we optimize up to 30 nanomolar, compound 13, and so improving more than 50,000 fold. And also we reverse completely the selectivity because this compound 13 is inactive towards CDs by testing through SPR. And with that, I would like to hand over back to Martin.
So with this tool compound in hand that had excellent potency towards DCAF1 as well as good selectivity against the previous known WD40 domain EED, we were wondering, is this a good molecule to tether to other warheads to turn this molecule into a PROTAC? So by SPR measurements, we saw that it has a long off rate, which is generally accepted that it's quite a good thing to have as your E3 ligand binder. And we also were wondering, do we have a good exit vector? So this crystal structure of compound 13 already suggested that we are quite close to the solvent. So we designed a PEG-linked variant of this compound termed compound 15, and we were able to solve the crystal structure of this. Shown on the right side in the left panel is the overlay with the compound 13, the parent compound, and you can see that PEG linker does not change the binding orientation, and we confirmed by SPR that also the affinity is not impaired by it. Recently, there was a great paper from the SGC that found another very potent ligand to DCAF1, and on the right side, the right panel, you can see the overlay of our compound versus the SGC compound. The SGC compound binds in a slightly different spot. It's further down the donut hole and is less solvent exposed, but has a very comparable affinity towards DCAF1. So this encouraged us on the next slide to test, well, we tested against one WD40 domain, but in order to have a good E3 ligand, we want to have selectivity preferentially across the entire proteome. So now, knowing that we have a valid exit vector, we now went to tether it to beads and do a chemoproteomic pull-down experiment, which you can see the data on the right. And we were very happy to see that we pulled out all the main parts of the of the CUL4 DCAF1 complex, as you can see. Needless to say, we didn't pull out EED, and we also didn't pull out any other very much unexpected off-target. So with this clean off-target profile, we've thought, okay, let's move it to the next stage. So we can go to the next slide. This is a short summary. So as Anna mentioned, we started really from a selection for an essential ligase, then doing SAR, we arrived at compound 13, and we tested that we have both an exit vector that doesn't compromise the affinity, as well as have a good selectivity for DCAF1 against other proteins. So finally, we decided, okay, we need to see if we can actually degrade proteins using this reversible DCAF1 ligand. And we chose BRD9 as a prototype. BRD9 for two reasons. We had like good ligands in hand, and published from BI, as well as BRD9 is a nuclear protein. DCAF1 is mainly expressed in the nucleus. So we felt this is it's a good starting point to test this. So we synthesized the molecule DBR1, tested ternary complex formation by SPR, showing the classical hook effect, and then went on to cellular tests where we could see a dose-dependent, time-dependent degradation of BRD9 that were rescued by inhibition of nedylation and the proteasome and E1 signal system. So new ligases always require that you do or you validate the pathway that your ligands actually work only due to the novel ligase. So in order to do that, we used our BRD9 degrader to establish the BRD9 rescue experiment using a CRISPR array library to actually test, are we on spot? Do we actually degrade our targets solemnly based on DCAF1? And we ran this focused ubiquitin focused CRISPR library of approximately 1,000 genes in this assay to see which knockout actually provides a rescue of BRD9 degradation. And as you can see, hopefully not too small in the left side here, DCAF1 was our top hit with other known interactors such as RBX1 also in the top list. Interesting, we also found a lot of proteasomal subunits unexpectedly, and even ubiquitin precursors. We then went further on and tested this with different guides just to double check that we were still not biased by this library. We saw that RBX1 as well as DCAF1 led to a complete rescue of BRD9 degradation. Interestingly, DDB1 showed less effect when upon knockout on BRD9 levels, and as well did CUL4A and 4B alone, but the combination actually improved a bit the rescue effect. So with this, we felt, okay, we have shown that we can degrade nuclear targets such as BRD9. It would be now interesting to see if you can use this novel ligase with this reversible binders also to degrade targets outside the nucleus. And therefore, we designed Dasatinib-based PROTACs. And we had an in-house compound that has a cereblon warhead to benchmark this. And we tested it on a couple of known Dasatinib targets in a Western blot experiment. And here you can see that our DCAF1-based degrader did indeed degrade, for example, CKS1 and LIMK2, but did not, unlike its cereblon counterpart, degrade ABL, for example. So that was quite interesting that we saw a different pattern. Further studies need to be done to elucidate this better, but we are currently working on some follow-up experiments there just to see, do we get an edge on selectivity using a different ligase, which would be very interesting. And on the next slide, we then performed a proteomics experiment just to see what other targets our DCAF1 degrader with a Dasatinib warhead can degrade. And we identified TEG, CKS1, LIMK2, TNK2 as kinase targets. ABL1, as indicated by the Western blot, was not affected as well as was LIN, which was quite interesting. And with this, we went further to test for BTK degradation. BTK is a clinical relevant target. Therefore, there was interest in using DCAF1 for potential degrader here as well. We had to switch the cell system because HEC cells do not really express well BTK. So we used a leukemia-dependent leukemia cell line that is dependent on BTK expression. It's called TMD8. And you could see that our Dasatinib-based DCAF1 degrader did indeed degrade BTK in these cells as well. And then we further used this TMD8 cell line to establish a high throughput BTK degradation assay based on a BTK GFP fusion over an M-cherry background that we then used in a FAX experiment to not only assess our degradation of BTK versus our M-cherry background, but also using the site scatter plot there to actually assess for cytotoxicity of our compounds that could confound our observations of degradation. And you can see that on the, there you go. You can see on the lower one that our dasatinib compound did indeed show toxicity in this system, but we got a window of roughly 10 to 13-fold of DC50 over GI50, while the BTKI inhibitor did not affect BTK levels but showed toxicity. So with that, we can go to the next slide. So with our promiscuous Dasatinib-based DCAF1 degrader in hand and the encouraging data that we can indeed degrade BTK, we thought, okay, can we get work on more selective BTK degraders using our Dasatinib reversible binders? Therefore, we started very simple. We fused an in-house BTK binder with simple PEG-linkers of different lengths with our DCAF1 binder, and we tested it in the aforementioned high throughput FAX assay. And as you can see, our short PEG-linked PROTAC molecules did indeed partially degrade BTK, but only up to around 50%. Interestingly, with a very long PEG-12 linker, we observed BTK degradation of roughly 100% with a DC50 in the low nanomolar range, and a quite good window in terms of toxicity in our artificial test system. So with this in hand, we went further on to decide, well, we had now a PEG-12 linker in hand. PEG-12 is not very drug-like and not very handy for any further studies. So we were thinking, can we test different, more complex linkers to establish protein PROTACs that have similar DC50s, but with a smaller molecular weight and better PK properties. Therefore, we designed 3 further PROTACs shown here on the left. The last one used a slightly altered BTK binder as well. And we tested them in the same assay system as shown on the right side. And interestingly, the shorter or more rigid linked versions, DBt6 and 8, showed again partial degradation, DBt8 a bit better than DBt6, but very, very gratifyingly, we saw that DBt-10 did indeed show a very similar DC50 compared to our PEG-12 linked version, with an excellent ratio of our GI50 versus DC50. So with this molecule in hand, we went further to test if we can still degrade BTK on endogenous level using the wild-type TMD8 cells. And we observed that we again have a time-dependent and dose-dependent degradation of BTK that can be again rescued by chemical inhibitors of the proteasomal pathway. So further on the next slide, and I'm about to wrap up. So now with a more or less potent BTK degrader that is not cereblon dependent in hand, we were thinking, can we actually use this on this novel ligase to rescue cereblon resistant cell lines that upon treatment with cereblon-based PROTACs do not degrade BTK anymore. So we created a TMD8 cereblon-resistant cell line. We tested our compounds head-to-head against a cereblon-based BTK degrader that was not functioning in these resistant cells anymore. And again, gratifyingly, we saw that our DCAF-based PROTAC could still degrade BTK, but the cereblon-based PROTACs did this not. And then we also tested the effects not only on BTK levels, but also on proliferation. And as you can see on the top left and on the right side, there was a massive difference between the wild type and the mutant cells in terms of proliferation upon treatment with the cereblon-based PROTAC, while there's no significant difference in our DCAF1 PROTAC shown on the bottom right in green. And with this, I will try to wrap up because we're close to time. So we identified new reversible binders to DCAF1 that have a high potency, have a suitable exit vector, have a good proteome-wide selectivity. We showcased that we can degrade nuclear targets as well as membrane bound targets, cytosolic targets, and we can use DCAF1 based PROTACs to overcome acquired cereblon resistance in leukemia cell lines, as shown on the right side. And with that, yeah, thank you for your attention, and hopefully there's time for some questions.